Good morning students. Today we are going to discuss about mass production of trichoderma and seed biopriming with trichoderma. In agriculture, worldwide pathogens are threat to crop production. The extensive use of fungicide in various parts of the world for years has increased pollution, increased serious water and food pollutions, and directly indirectly it is affect to the human health. Apart from this, the chemical tend to become less efficient due to the development of the resistance among all the pathogens. Hence, it is necessary to look after for the other management practices, which include the use of the eco-friendly biological control agents. Now we are going to discuss about the history of the trichoderma. In 1671, the first found in Germany. Then after in 1794 identified by the Pearson, which is 218 years ago. Then after Gilman and Abbott recognized 4 species of the trichoderma according to their color, shape of conidia and colony appearance. Then after Wending first demonstrate the parasitic activity in wilt of pigeon pea. Now we are going to discuss about the main characteristic of this fungi trichoderma. The main characteristic of this trichoderma fungi are free living, very effective biological agent, ubiquitous in nature, non-pollutive, easily assessed, non-phytotoxic, cost effective, longer shelf life and growth greater compatibility. These all are the main characteristics of the fungi which has capacity to control the diseases. Then after, which type of temperature is required for the growth of the trichoderma fungi? Basically 25 to 30 degrees centigrade temperatures are required for the growth of the fungi, but some of the trichodermas are there which has capacity to produce their growth at 45 degrees centigrade temperature. There are 2 media for the growth of the fungus. The first one is a cornmeal starch agar media and second one is a potato dextrose agar media. Potato dextrose agar media is generally using for the growth of this fungi. In potato dextrose agar media, the colony is white in nature. Then after, a yellow pigment is secreted into the agar, especially in PDA media. Some species produce a characteristic sweet and coconut odor. Then after, which type of mechanisms are there for the biological control and which is the mechanism for control of the disease? Various types of mechanisms are there. For example, antibiosis, parasitism, inducing host plants resistance and competition. Among that, we are going to discuss about the antibiosis. And antibiosis, trichoderma produces various types of chemicals, trichodermin, viridin, trichosacin. These all are the chemicals which are produced by the trichoderma for the control of the disease. For example, in Rhizoctonia soloni, the chemical which are produced is the gliotoxin. These are the chemicals which are produced in the Rhizoctonia soloni disease for the control of the disease. Biocontrol agents are from the species Trichoderma aspirin, Trichoderma hercerium and Trichoderma viridi. These biocontrol agents generally grows in its natural habitat on the root surface and so affects root diseases in particular, but can also be effective against foliar diseases. Trichoderma viridi is a fungus and a biofungicide. It is used for seed and soil treatment for suppression of the various diseases caused by fungal pathogens. Trichoderma, a filamentous fungus, is opportunistic or virulent symbiotes that are used as a biopesticide, biofertilizer or fertility promoter worldwide. Application of trichoderma increasing the plant growth, yield and increasing nutrient availability of the plant. It is an excellent growth promoter which solubilizes the phosphorus and micronutrients. Application of the trichoderma strain in rhizosphere of the plant increasing the number of deep roots, thereby increasing the plant's ability to resist drought. So various advantages of the trichoderma are yield increasing, germination rate increasing, root length and shoot length increasing, solubility of various insoluble phosphates are increasing, augment nitrogen fixing, dry matter increasing, act against wide range of pathogen fungi, growth rapidity and colonize the soil. Naturally long-term immunity to crop and soil. So what are the disadvantages of the using of the trichoderma? Harmful parasitic of the mushrooms loses its efficiency if not placed in its natural condition. It cannot be used as foliar spray. Don't grow in an alkaline pH. What should be the precaution we are handling when we are handling to the trichoderma? Don't use chemical fungicides after application of the trichoderma at 4 to 5 days. Not use trichoderma in dry soil. Don't put treated seed in direct sun rays. Don't keep the treated FIM for longer durations. Seed biopriming varu.
Now coming to the media preparation for further mass multiplication of trichoderma. Here all the equipments are listed. Also the materials are given. The spatula, potato dextrose broth media, potato dextrose agar media, aluminium foil, steel vessel, glass rod, measuring cylinder to measure the required quantity of water, funnel to pour the media into the flask, flasks as per the media to be prepared. Here we have taken 500 ml and 1000 ml flask. An antibiotic, streptocycline, non-absorbent cotton, and the rubber bands if your flask does not have a cap. For 500 ml of PDA, we will need 39 gram of PDA, and for 500 ml of PDB, we will need 24 gram of ready-made media, as we are weighing here. Now, take the required amount of the water in a clean vessel. Start the gas stove. Add the water in it. Now, add the weighed media in the vessel. Make sure you wear the gloves during entire process. Mix the media with glass rod simultaneously so it doesn't stick to the bottom of the vessel. Also, it helps in dissolving. After complete mixing, add the media into a sterilized flask using the funnel. Make sure you do not fill the whole flask. Flask should be filled half up to its capacity. So autoclave process can be smooth. If you are adding antibiotic which is a heat resistant, then you can add at this stage. And if you are adding the streptocycline as we are doing here, you can add it after the sterilization of the media. while pouring. Now here, this is the horizontal autoclave. It is highly advanced machine for sterilization purpose, for moist heat sterilization. Put the media inside it. For 121 degree centigrade, 15 psi, up to 21 minutes. This autoclave helps to sterilize the area of the inside of any vessel, whichever we keep aside. Carefully keep aside all the flasks containing PDA and PDB. Close the lid of autoclave and set it. Thank you. Now we will see about the mass multiplication of Trichoderma. First, start from the cleaning the laminar airflow. Sterilize your necessary equipments. Sterilize the wire loop, forceps, and cork borer using flame sterilization. Now take a sterilized broth side by side. As we all know, Trichoderma is a cheap, very easy to use and eco-friendly. It can be used for integrated disease management. Now add the streptocycline, which is an antibiotic. Here we add it after media sterilization and media is cooled a little bit up to room temperature, as the streptocycline is a heat sensitive antibiotic. We are not adding before autoclaving of the media. Follow the same process with the second flask containing PDB media. Care should be taken while stirring the flask that air bubble should not be formed. Here you can see how the person is mixing. Now take a culture plate of Trichoderma viridi. Cut the 5 mm disc from the culture plate of Trichoderma using cork borer. Make sure you flame sterilize it after each usage to avoid any contamination. Then clean and sanitize all the area. Now take a forceps, lift the disc and put it in a broth. As we all know, mainly Trichoderma has 3 main mode of action. First is antibiosis, second is mycoparasitism, third is competition, and fourth is plant growth promoting activities. To conclude it, there are 4 main mode of action. This can be found, the Trichoderma can be found generally in the rhizosphere of a plant. In our department of pathology at AUN, we grow generally Trichoderma viridi, and mass multiplication also occurs for the species Trichoderma viridi only. Clean and sanitize every equipment after each usage. This ensures a proper contamination-free practice. Now tie them and put them for 3 to 5 days in incubation for 22 to 25 degrees centigrade in an incubation chamber. See, for mass multiplication, we generally use the trays and we add the prepared broth which we have earlier shown after it has the growth of Trichoderma viride. First, clean the tray properly using cotton swab and alcohol along with the all side. Now, add the prepared broth which is having Trichoderma culture. Here we fill tray up to half, but to show we are generally using earlier which we have used PDB. Now cover the tray with aluminium foil properly and seal all the sides of the tray using aluminium foil. If foil breaks, make sure you cut the piece of foil and seal the all sides of tray. This prevents any contamination and further helps to grow the trichoderma layer on the broth. Seal the tray very well. After sealing, now we will keep the tray on a wooden rack in a controlled temperature room on a wooden rack for further growth and multiplication. This area should be clean. Here are the glimpses of the ALP students while working on the trichoderma mass multiplication. Now we will know about the biopriming and seed biopriming. Seed priming is controlled hydration technique in which seeds are soaked in water to a point where germination related metabolic activities begin in the seed, but radical emergence does not occur. This in the video you can see the Anubhav brand Trichoderma viridi, which we will use further for biopriming process. Biopriming is a technique of seed treatment that integrates biological, which means inoculating seed with the beneficial organism, and physiological, which means seed hydration aspects of the disease control. Blotter paper, plastic tub, the seeds which are to be treated, water bottle, this spatula, this all the equipments we will need for seed biopriming. First, take the clean plastic tub. Make sure you wear gloves before performing this operation. Now add the water little by little. We have taken the required quantity of bioagent as per shown in the packet for our seeds. Now make a slurry using the clean water. Mix it properly to avoid any lump. This technique helps the plant to activate defense response more quickly and effectively. At the same time, it helps the plants to grow efficiently. Now, add the seed, mix it properly, coat them using the slurry. Make sure to coat each and every seed with the bioagent slurry by proper mixing, as you can see in the video. This technique of seed biopriming is based on the controlled hydration of the seed. This Bioagent Trichoderma viridi has contained CFU of 2 into 10 raised to 8. We are taking good time for mixing it, so each and every seed is coated properly with bioagent slurry. Now take a blotter paper, or you can use a clean cotton cloth. Spread the treated seed evenly. Make sure you shade dry the seeds. Spread it evenly to soak up the extra moisture and proper drying. Do not put this treated seed into the direct sunlight. Place them in a shady area. Now these seeds should be kept for 2 to 3 hours for drying purpose, and on the next day these seeds can be ready for sowing purpose. You can see each and every seed is well coated. With bioagents. Now coming to the more information of trichoderma. fungi. First of all, it is a biocontrol agent. So what are the basically biocontrol agents? According to the definition given by Becker and Cook, first we will know biological control. So biological control is a reduction of inoculum or disease producing activity of a pathogen accomplished by or through one or more organisms other than the man. In this particularly biological control, whatever we are using is biocontrol agent, such as antagonistic microorganism, Trichoderma species, Penicillium species, Bacillus and Pseudomonas bacteria. Here you can see this is the Trichoderma, this is the Penicillium, and this bacteria are Bacillus, and this one is Pseudomonas. Now, the features of Trichoderma, they are very effective biological agent till date. They are free living, they are ubiquitous, everywhere, highly proliferating, they are non-pollutive, easily accessible, also they are non-phytotoxic, systemic, ephemeral, they are readily biodegradable, cost effective, main feature, synergistic effect, also main feature, longer shelf life, and greater compatibility with other biocontrol agents. Moving to the history of Trichoderma, it was first found in Germany 1671. In 1794, identified by the person, and in 1927, Gilman and Abbott recognized 4 species based on their color, shape of conidia, and colony appearance. Also, the potential use of Trichoderma by Winling, and he was the first to demonstrate the parasitic activity in wilt of pigeon. Based on mycoparasite against many soil-borne plants. It's a free-living fungi common in soil and root ecosystem. They are highly interactive in root, soil, and foliar environment, rhizosphere and planosphere. It suppresses the pathogen by different mechanisms of biocontrol. Here are some of the general characters. They require 25 to 30 degrees centigrade temperature for fast growing. The conidia forms within a week in compact or loose tuft in shade of green or yellow, or less frequently white. Yellow pigment may be secreted into an agar, especially on PDA, that is potato dextrose agar. They have particular smell which can be described using words sweet or coconutty odor. Here in the first A, you can see Trichoderma on PDA plate as well as agar. Slant. B is a microscopic view, but which is not more clear, but further we will see clear pictures. In C, it is in the broth. This is the morphological structure of Trichoderma. As you can see here, these are conidiophores on which conidia bears, and phyalids, also the hyphae. These are all the different colonies of different Trichoderma species on PDA plate, dorsal view from the upper side. From the right side, 1A and 1B, as you can see here. These are the colonies of Trichoderma strains, different strains of Trichoderma on this plate. and their conidiophores with their conidia. This is the Trichoderma pseudoconingi with the microphotograph. You can see their conidial shape differentiation by comparing these photos, microphotographs. Now third is Trichoderma harzianum. Fourth one is also Trichoderma harzianum. And fifth one is also Trichoderma harzianum, but with different strains. Now, their conidiophores are highly branched, as you can see here. There are so many branches. They are generally loosely tufted or maybe compact tufted. The main branch of conidiophore, this produce lateral side branches. The branches may rebranch with the secondary branches and longest secondary branches being close to the main axis. This is the stereomicroscopic view of a Trichoderma harzianum. Their conidia typically appear dry, but in some species they may be held in a drops of clear green or yellow liquid. For example, Trichoderma virens and Trichoderma flavofuscum. They are generally round to oval shape, as you can see in previous pictures also. Generally round to oval. This is the hypocrea telomorph of the Trichoderma species. This here you can see the mature perithecia. Here teleomorph means the sexual reproductive stage of fungi. This is the very close view. This is the mature perithecia. Spores will get released from this side. Where we can find Trichoderma? So they can be easily isolated from soil, root and decaying wood also. Other forms also which contains organic matter. This is the potential biocontrol activities which Trichoderma possess. First is antibiosis, antibiotics production. We will see all of these further. Second is mycoparasitism. Third is systematically acquired resistance, competition, growth promotion, effective antagonist, rapid substrate colonization, and enzyme production. Now competition. They compete for space and nutrients with other fungi and other phytopathogenic fungi. They suppress the growth of pathogen population by doing so. For example, soil treatment with Trichoderma harzianum spore suppressed the infestation of Fusarium oxysporum forma species Vesinfectum and Fusarium oxysporum forma species Melonis. Mycoparasitism. Mycoparasitism is an antagonist fungi parasitize the other pathogenic fungi. Here in picture you can see the hyphae of this is the hyphae of Trichoderma and this thick hyphae is of phytopathogenic fungi. Hyphae of Trichoderma either grow along the host hyphae or coil around it, as this picture shows. For example, Trichoderma harzianum and hamatum are mycoparasite of both Sclerotium rolfasi and Rhizoctonia solani. What they do interact? They coil the hyphae around the pathogen, their hyphae around the pathogen. They vacuolize and they penetrate by making hostoria, and finally they lysis the host. They promote the lysis of host by enzyme segregation, like beta-1,3 glucanase, chitinase, protease, lipase. Trichoderma coils around, penetrates and kill other fungi that are phytopathogenic to crops. This is a clear view within an electron microscope. Here you can clearly see the mycoparasitism action performed by Trichoderma, the enzyme secretion. Antibiosis. So the antibiosis is a condition in which one or more metabolites excreted by one organism has the harmful effect on another organism. Trichoderma secretes trichodermine, viridine, trichothecene, and sesquiterpene, and many more. See, this is the growth inhibition of Rhizoctonia solani by the Trichoderma virens produce antibiotic gilotoxin. This is the area gilotoxin amended, where here it is not amended. It also works as plant growth promoter. Some of Trichoderma strains solubilize the phosphates and micronutrients, which helps the plants to absorb the more nutrients from the rhizosphere by increasing the number of roots, deep roots. Also, it's increased the ability to resist the drought. It increased the ability of a plant to resist drought. In picture, you can see enhanced root development in T22 with T22 and without the treatment. So how we can isolate the Trichoderma from soil? So isolation from soil on specific media if required. Then incubate 7 days at 25 degrees centigrade. Subculture them on PD plates. Purify them. Inoculation of purified culture into the slants. And preserve those slants in a deep freezer for further study purpose. Now coming to the mass production using liquid fermentation method. So you have to mix 30 grams of molasses and 6 grams of brewer's yeast in 1 liter of water. Distribute 60 ml of this mixture in each conical flask. Autoclave them. Inoculate 8 mm micellar disc of Trichoderma in this media. Incubate for 10 days at room temperature only. Use for the multiplication in the fermenter. Prepare 50 liter of molasses plus yeast media and sterilize them for 30 minutes in the fermenter itself. Now transfer aseptically 1 liter of Trichoderma. Then again incubate it for 10 days using the hemocytometer. Colony should be count. Colony forming units, for yawning. Colony forming units should be count. 500 ml of fungal biomass plus 1 kg of talc powder should be added. Then add dry. Add the carboxymethyl cellulose and sticker 5 gram. Store in polythene bag. Here are some substrates for the mass multiplication. This is a wheat bran, wheat straws, FYM, pressed mud, coconut coir paste, groundnut shell, and rice bran. Now carrier material. It can be talc, vermiculite, molasses, gypsum, kaolin clay, cocoa peat or what peat, normal peat, sawdust, sodium, alginate, CaCl2, that is calcium chloride. Now looking to the advantages, this and trichoderma enhances yield along with the quality of produce. They boost germination rate also if we are giving them seed treatment. They increase in the shoot and root length as well. They solubilize various insoluble forms of phosphates and other micronutrients. They helps in nitrogen fixing. They promote the healthy growth in early stage of crop so the disease can be escaped. They increase the dry matter production substantially. They are harmless to human and livestock. They act against wide variety of pathogenic fungi, phytopathogenic fungi. They perpetuate themselves by producing an ample spores. They grow rapidly and quickly colonizes. They can promote nutrient uptake and enhance the plant growth. They provide natural long-term immunity to crops and makes the soil texture better. Now disadvantages. So they are very harmful for mushroom production. We may call it the trichoderma as a weed for mushroom production. They loses their effectivity if not placed in their native condition. It's not effective as foliar spray. It does not grow in alkaline pH above 8. They are generally zone specific and having slow growth also. These are the different methods by which you can apply this bioagent. First is the seed treatment. Mix the talc formulation per kg of seed 6 to 10 gram before sowing. Nursery treatment, cutting and seedling root dip method. In that, mix 10 gram of trichoderma powder along with 100 gram of well rotten FIM per liter of water and dip the cuttings and seedlings for 10 minutes before planting. Soil treatment. The plant treatment, branching near the soil and stem region that we generally call rhizosphere, wound application and furrow application. See, these are some precautions which should be taken while dealing with the trichoderma. See, don't use chemical fungicide after application of trichoderma for 4 to 5 days. As these spores are alive and trichoderma also is a fungi. So we are not going to kill the fungi which is beneficial for our plants. Do not use this trichoderma in dry soil. Moisture is must to let it work and survive. Don't put the treated seed in direct sun rays. Make sure you shadow dry the treated seed. Also don't keep this treated FIM for longer duration. Now if we see the compatibility of trichoderma, they are compatible with organic manure, biofertilizers like rhizobium, aspergillus, mycorrhiza, azotobacter, bacillus, phosphobacteria. All the bacteria and ectomycorrhiza. It is compatible. Now, trichoderma can be applied to seeds treated with metallixyl or captan or the carboxyl, carbendazim, not the mercurial ones. So here you can see the compatibility test between Trichoderma viridi and Pseudomonas fluorescens. So here are several reputable companies that manufacture government registered products. These are the products name: Ecoderma, Bioderma, Tricovine, Biohit, Monitor, Bestderma, and TricoXP. They come in a variety of formulation: powder formulation, encapsulation in organic polymer like sodium alginate, spray form, emulsifiable concentrates, molasses, enriched clay granules. These are all available in worldwide. Pelleting biomass and bran with sodium alginate. To conclude this topic, I would like to say the use of trichoderma has gained importance in managing most of the plant pathogens. However, there is still considerable interest in finding more efficient mycoparasitic fungi, especially within Trichoderma harzinum strains, which differ with respect to their biocontrol effectiveness. The technique for mass production and use of these bioagents have been commercialized for the purpose of producer as well as farmers. Thanking you.